But I've known Mary almost since you've gotten here, I think, and had her in a lot of classes. And as I was sharing with some other people, as a faculty member, we get a chance to really see how students grow on their journey here. And she's going to leave a lasting impression with me forever. And so before I get all emotional, I will read her autobiography. Mary Helms is the daughter of Don and Julie Helms of Arlington, Nebraska. She graduated from Arlington High School and is majoring in biology, obtaining a minor in public and global health, and will graduate in a week. While at Wayne State College, she has been part of the Wayne State College cheer team, CSF, chess club, Spanish club, student senate, and Phi Kappa Phi Honor Society. She is also working as a navigator, peer tutor, supplemental instructor on campus, and after graduation, she plans to pursue a PhD in immunology to further her career in biomedical research. Can I add a few details? She is also going to leave right after graduation to go to Nepal and do research in Nepal with a very innovative Wayne State College alum, Dr. Devesh Karmacharya. And so she'll be spending, what, 3 weeks there?
Yeah, 3 or 4 weeks.
Yes, and I really regret that I'm not able to be there while she's there, but we'll do it again another time. And then in September, I think you will be going to Spain for a Fulbright English teaching assistantship. She is going to grow and make a difference in a lot of people's lives while she's doing all these things. But when she does go back to her PhD research, she's going to bring a perspective that's going to wow that lab to whichever lab you go to. And yes, last night she just received the Ron Holt Civic Engagement Award too, as if these are honors. I mean, she's right up there with everyone. So she is going to tell you about investigating the role of HCMV, US-18, and viral infections, the cytomegalovirus infections. So thank you.
Well, thank you so much for that wonderful introduction, Dr. Ingebrigtsen. Yes, I cannot wait to tell you about investigating the role of gene US-18 in human cytomegalovirus infection. However, I understand that a lot of us are here to support me professionally and personally, not because you just love human cytomegalovirus. So let me explain why I got here and what that is really quickly. There we go. Okay, so I started researching in the summer of 2022 at UNL. My advisor, Dr. Christensen, said there's the summer program, I think it'd be of interest to you. And so I got started there. I worked full-time in the lab. We lived with my cohort of about 100 different students, all researching different things. And I just fell in love with research, just being able to push our understanding of the biological world, really, really neat. And I was placed in the virology center. And I hadn't had much experience with that before, but through the Crawford lab, I learned so much. And she studies human cytomegalovirus. That's me presenting my research, where's the pointer? At the end of the year symposium. So let me tell you about human cytomegalovirus. So that is strain 5 herpes virus. We see it listed among the 8 common herpes viruses there that are commonly known to infect humans. And so when you think of herpes viruses, there's a lot of different things that might come to mind. But with human cytomegalovirus, or HCMV, it's easier to say, that's what I'll call it from now on, HCMV infects 44 to 99% of the population. So actually the majority of people are infected already and don't know it. It is passed from mother to child along with a lot of other different ways. And so it's pretty prevalent in our society. This varies though, the range of how many people are infected varies based on geographical location, age, gender, socioeconomic status. But we do see that when it does infect people, it establishes a lifelong infection. Just like most herpes viruses, it's going to be having processes of latency and lytic stages that I'll get into more later. And even though we don't see a lot of symptoms, people don't present, it's often subclinical, it still can cause a high burden of disease. So in people who are immune sufficient, we don't see a lot of that, but people who are on immune suppressants, those who are undergoing transplants, things like that, we see that it can reactivate and cause a lot of issues there. And then it is also the leading cause of congenital birth defects. So that's a pretty big deal as well that not a lot of people know about. So it's important that we understand the virus and this process of latency and reactivation, which let me tell you about that. Latent is that idea of like the virus is laying dormant in the cell. It's not really doing anything, it's just resting, not actively dividing. And then lytic is when it's actively replicating, it's able to be identified by the immune system, and so then it's able to be attacked. But when it's just hiding out in the latent phase, it isn't really doing anything, and we don't expect to see a lot of gene expression. If you don't know why that's important, I'll come back to that. But when it is there, it establishes latency in hematopoietic progenitor cells in the body. So let me walk through, where did I put this? There we go, the process there. So herpes virus is going to enter the cell, and this is for all types of herpes viruses, not specifically HCMV, but it enters the cell there, and then it's going to uncoat, and then it has the viral DNA that can then go to the nucleus and go and interact with our own host DNA, and it can also then go to lytic infection. However, it also is able to, once it's in the host chromosome, it can reactivate, so that it goes back to that active replication. And so this process specifically, this reactivation from latency, is what the Crawford lab was most interested in, and where my research interests therefore lay as well. And so we wanted to find a gene that was expressed during latency. And so, there we go. Up there, okay, thank you. And so my project was wanting to find a gene that was expressed during latency. And so for my non-biology friends, gene expression is when we have the DNA of the virus of a human, and we have different genes that make up that DNA. In HCMV, there are 200 genes that make up its entire genome, and so then one of these genes can be turned into RNA, a single strand of RNA, and then turned into an amino acid chain that will fold into a protein. And that whole process is what we mean when we say that a gene is expressed. So if it's latent, laying dormant, we wouldn't expect DNA to be doing that. We would just expect it to stay DNA. However, my gene of interest, US18 is what it's called, goes through this process even during latency. So that was really interesting to us, and we wanted to figure out why. So we found, looking at previous literature, if you look down here, this is US18 highlighted among the US12 gene family. And so it is expressed during latency, we see those high bars, whereas these other genes, really we don't see a lot of expression during latency. That's how we ended up deciding to study this. And what we know about US18 from previous research isn't much. It's not highly studied. We do expect it to be a G protein coupled receptor. We see that in the corner there. We expect that because of the 7 hydrophobic regions found within the protein domain that we would expect to then embed in the phospholipid bilayer membrane. It's also expected to interact with the Rho-GTPase signaling pathway, so we think that it might be related to cell growth and proliferation. So, for my non-bio friends again, stay with me. Basically, we're interested in the US18 because it's expressed when other genes aren't. Okay, so my research goals were to study it more, understand why is it being expressed, what is it doing. And so we want to create tools to be able to do so. And so we want to amplify the gene and put it into a plasmid that can replicate in human cells so we can study it there. Pretty straightforward on paper, not so much in real life. So the process of gene cloning overview, let me tell you, it's normally, if we wanted it to look like this, it didn't actually, but we wanted to amplify the gene, so just make a lot of copies of the gene so we have stuff to work with. Make sure that we made the right thing by checking the size. Then we wanted to purify it, so we would just get the DNA, so we could work with that. Then we wanted to put it with a plasmid and put it into bacteria, just so that it's more stable. We can't just go put like a single gene into human cells. It would get chewed up, it would not be able to replicate on its own, so we have to put it into a vector that can be more stable and do that for us. So that is the basic idea of what we were trying to do. And so step one, this is all I did at UNL originally, and so we amplified it, and we used polymerase chain reaction, or PCR, which is using different temperatures to just create a bunch of copies of the gene from the template. And so we see the DNA coming apart, then we use it as a template. We have primers, these little pieces that are going to tell the machinery where to start making new DNA so that we just get our gene that we want. So we're not going to copy the entire virus, we just want the one gene. So I designed primers that would then tell that machinery where to start and where to end. And so in those primers, we put in restriction enzyme cut sites. So think of that as like a site that's telling some really specific scissors where to go then later cut the DNA, because that's important. But right now, we just put that in thinking ahead. We need to cut it later. This is how it knows where to cut it. And then we just did that a bunch of times. And then we went to go see if we actually did it. And we checked the size using gel electrophoresis that separates the bands by size. Indicated by this red arrow, we see that we got a very light band, around 825 base pairs, which is what we wanted. However, also in this lane, we see these non-specific bands that we didn't want. That was not the goal. And so we did a lot of troubleshooting trying to get rid of those, just using different concentrations, adding different things. And we did that over and over and over again for the majority of my summer research program. However, we never got rid of those bands, and we eventually just decided to move on, because there was a way to move on even with the non-specific binding. And that was through cutting out just that very light band. We actually just went in with like a little knife and cut it out of the physical gel, and then went to purify it so we got rid of the gel, so we would in theory just be left with our gene of interest, with the DNA. And so then we moved on. We wanted to ligate it with a plasmid. So remember I said we had those like cut sites that were going to tell the scissors where to cut. And so that is here, those yellow sites, is going to tell the restriction enzymes where to come in and cut very specifically in the DNA. And that's going to line up with this circular bacterial plasmid, which remember that's to help with stability and so it can replicate itself. That's going to line up there, so they ligate together, and then ligase is going to make that whole thing circular so we can then go put it into bacteria cells. It's trying to. There we go. And so transformation, that's that process of putting the plasmid, the vector, and that insert that we just made into bacteria cells using a heat shock that would open the membrane of that bacteria cell for just a little bit so that the plasmids that we wanted could get into the bacteria cell and then be reproduced by that bacteria. We hope. Hypothetically, this is what we thought happened. However, we never checked that. I ran out of time with a lot of troubleshooting and everything. That was the end of my summer program. They said, pack up, it's time to go back to school now. And so to overview everything that I did at UNL, we were able to get bands to show up, gel purify them, and we think put them into our DNA, the DNA into a vector and then put that into bacteria. But again, never double checked that, and it maybe wasn't the most ideal way. So I wasn't ready to give up on this project. I was still really interested, and so with the help of Dr. Christensen and my mentor, before we moved this to Wayne State College for me to continue working on. And so we tried a whole new PCR approach before we put on those restriction enzyme cut sites that extended our primers. We thought that maybe that was leading to the non-specific binding, so we just got rid of that. We designed new primers without those. And so then we got a band around 825 base pairs, right where we wanted, and that was awesome. We got the single band, it was right where we wanted, we thought, we did it, this is great. And so we moved forward. However, the next step would have been gel purification. Other people in our lab at the time, after I gel purified, were not getting good results with the gel purification. We had used a different intercalating agent, and we used cyber green here instead of ethidium bromide for my non-bio friends. You don't have to worry about that, but we thought perhaps the different intercalating agents is what was causing those issues. So we just decided to bypass the gel purification altogether and move on to a different route, and that is to DNA or to purify the PCR product directly. So we ran that gene through that thermocycler to get a lot of copies, remember, and so now we just needed to get rid of all the extra stuff, so we'd just be left with the purified DNA, so we're getting rid of the extra base pairs and everything like that, and should be just left with the ready-to-use gene of interest, US18, and so then we have that product ready to ligate. However, if you remember me saying, we took out those restriction enzyme sites, so we now didn't have somewhere to tell enzymes to cut. So we used a new method, and so this is using a new vector called PGMT, and it has T overhangs on it, and so that it would ligate with US18. Taq polymerase is the polymerase we use during PCR, and it's known to add extra A overhangs to the gene of interest. And so the A overhangs would then be added to US18 and then go with the T overhangs, because in DNA, A's and T's go together. And so then this should ligate our vector into one big thing with our vector and insert all together, we hope. And so then we moved on and we plated it with E. coli, that same process I showed before, where we open up the membrane, we put in our vector, and then the membrane closes, and it should be in bacteria. And that is essentially where we are at right now with that research. So we were in the process of verifying that that's exactly what we had. You go in and you just make sure that if you cut it in certain spots, that you would get the right length of bands with the insert. And if you don't have the insert, it won't show up when you run that gel electrophoresis to see how long the bands are. So that's where we're at right now. And so, in conclusion, science isn't always straightforward. When I originally put together my first draft of this presentation, and I was going to present it at NAS, at the Nebraska Academy of Sciences, the other weekend, I showed it to Dr. Christensen, and he said, well, we should add in more things, because originally I had put, here's what we're studying, and here's where we're at right now. And I didn't include all the troubleshooting and the, oh, we failed and we tried this and this wasn't working, so we tried this, because I didn't want to show people that I didn't do perfect science. But he said that's what science is. Science isn't always straightforward, and that's what you did. You did do science. All the effort and the trying, that was science. So what we wanted to do was this. We wanted to amplify it, check the size, gel purify, ligate, and move on with our lives. But at UNL, what happened was I amplified it, I checked the size, it wasn't what I wanted, so I amplified it again, checked it, and did that over and over and over again. And at the end of the summer, we did these steps really quickly and never confirmed if that's what we had. So then we moved to Wayne State, and then we amplified the gene, checked the size, and that was good. And then we moved on to PCR purification to bypass the gel purification. And then we ligated it with the plasmid, transformed it into bacteria, and that's what we think we have right now. And then we went to, in the future, isolate the DNA, and then ligate that with a new plasmid that can then reproduce in human cells. The goal is to get the, the transformed plasmid with our gene of interest into a human cell, so that then that human cell will express US18, And it's supposed to be a G protein coupled receptor, so we would expect it to show up on the surface of the cell. And then we'd want to see how that interacts during viral infection. Like, we want to figure out what it's doing. I don't have time for that right now, unfortunately. I'm graduating here soon. But other people are interested in this work, and we'll continue it hopefully at the Crawford Lab one day, because this is really interesting. But I've learned that I really love science and love research, even with all the troubleshooting and when things don't go exactly how I hope. And so yeah, I'm really thankful to have had these experiences. I want to acknowledge the University of Nebraska-Lincoln, where I started this research, especially the Crawford Lab. Dr. Crawford and our lab tech, Cameron Fenning, were a huge help in my learning process. And then everyone through the undergraduate summer research experience in neurology was so helpful. And then huge thanks to Wayne State College for letting me move this project here. Thank you, Dr. Christensen, for helping me through that process, for being the one to say, hey, I think you should check out the summer research program at UNL. And then also thanks to my funding from the National Institute of Health. And also thanks to you guys for coming to see me, even though you probably don't have a ton of interest in human cytomegalovirus. I really appreciate you coming and listening to me talk about it for a while. But my future directions, like Dr. E said, I'm going to the Center for Molecular Dynamics in DePaul. Super excited about that. Can't wait to learn everything that they have to teach me. And then complete a Fulbright grant in La Rioja, Spain. And then after that, I'm looking to pursue a PhD in immunology. So yeah, there's my references. And I'll take any questions you have. Thank you. Okay, yeah.
How long does the entire process take to take it apart, put it in the gel, do all that stuff?
Yeah, well, just the process of PCR and checking the size and gel electrophoresis, you can run that all in one day. In theory, you could probably do the whole thing in a week if everything went to plan. But here I am 2 years later and everything didn't go to plan. But I also am no longer doing it full time. But in theory, you could do it all within a week.
Well, kudos to Dr. Christensen for knowing how to skip that step.
Yeah.
But you've done a wonderful job, and I think that the fact that disappointments in the lab have increased your interest in becoming a scientist is a good sign. I have a question because I'm a physiologist. Why? So the question is, what cells, human cells, are infected by the cytomegalovirus, and why do you want to be able to isolate a latent gene for future diagnosis, detection, and what are your purposes?
Yeah, for sure. That's a really good question. So originally, what cells does infects a whole bunch of different cells during lytic infection. However, when it's establishing that latent infection, the reservoirs that it uses is hematopoietic progenitor cells. So those are immune cells, and so that's really interesting as well. But then when it is expressing this in latency, then since those are latent reservoirs, we'd expect US18 to be on the surface of hematopoietic progenitor cells. And so why this one? Well, if it's expressed during latency, we think maybe it's doing something with that reactivation. Maybe it needs US18 to reactivate, or it's in some way being used. And then also, and this is just my ideas, I think that it's cool that it's on the surface. G protein coupled receptors are known for being drug targets since they're on the surface, different things like that, so drug targets, possible vaccines, things like that. But that's way in the future, but just thinking, yeah.
Very good, thank you.
So to add to that though, so you're hoping to interrupt that process somehow to prevent it from causing the disease, or
Yeah.
you mentioned birth?
Yeah, so right now, first we have to figure out why. So this is a lot of just the figuring out what is actually happening in that process, just because so much is unknown right now. And then, but eventually, yes, figuring out why will help us then develop treatments, cures, diagnosis, things like that, yeah.
You said when you began that a lot of times it's passed from mother to child, but how are some of the other ways that
Yeah.
passed?
So it can be passed mother to child, like during pregnancy, during birth, breastfeeding. Also, it is a sexually transmitted disease, like we think of herpes viruses, but it can be passed through saliva and many other things like that. But it's interesting. Yeah, no, it's really interesting, yeah. And leads to, yeah, like we saw 44 to 99% of the population being infected. What is it?
To the aspiring researcher, what's like the general education?
General wisdom? Oh goodness. I don't know if I have any of that, but I guess for me, it was just like learning to love the process. Because as you saw, it was a lot of process and not so much results. But I still learned to love, like, I love going to lab and being like, oh, shoot, another roadblock, but that's just another problem to learn how to overcome. Thankfully, I've had a lot of great mentors who are like, here's how we overcome that, since I'm not quite to that step. But learning to love that has been really awesome. So I guess that's what I would say to an aspiring researcher.
Yes.
Sorry I came in late, but I wanted to also say that for those of you that don't know, troubleshooting in science is learning. So what you saw here is very typical. It would be highly unusual that everything worked right away. And two, Mary was my student that got away from Embry. I really wanted her in Embry. It was a 2-year program. And she's one of the few that got away from me. But I'm glad she's still going to that school.
Curious about the epigenomic latency. Do you,
Oh,
is that
yeah,
on the
let me
host?
go
Is that
back
on the
to
host
that.
gene?
So that's actually, are you,
Way back to the
yeah, let's see here. Here, are
yeah,
you
there,
asking about this?
The viral epigenome.
So some viruses will actually insert their DNA just directly into human chromosomes, which can cause lots of issues, that can cause cancers, things like that. But we don't actually know if HCMV integrates into the genome. We know it goes to the nucleus, so I mean, I would assume so. But I don't know if there's been actual research that's like, we for sure know this is happening and this is how it's happening.
Just sort of plug in for Mary as well. She will be presenting more science at 2 o'clock in Carhartt BV as she's got her poster, senior poster.
Yes, and that's over a whole new topic.
So it's like, she hasn't done enough science yet.
If you want to learn about using microscopy to study cilia and primary cilia dyskinesia, come check it out too. Thank you.
So are you thinking virology for your PhD, or are you still open to learning where you want to go?
Yeah, I'm so open to everything, which makes deciding really hard, which I'm hoping to figure that out in my year or two before I jump into grad school. But virology is definitely a top interest of mine. It's just, yeah, it's so cool and so prevalent to human health. Very important topic.